Hello. For the next 5 minutes or so, I'd like to talk to you about protein denaturation. But first, we need to talk about protein structure. There are 4 levels of protein structure. There is primary structure, which is peptide bonds between the essential amino acids that form chains. There's also secondary structures, when these chains fold up upon themselves and form alpha helixes and beta sheets. These structures are formed via hydrogen bonds between the different amino acids in the chain. Tertiary structure is when you get salt bridges and disulfide bonds between the amino acids in the chain. And these tertiary structures form a glob usually. Like they don't form long lines like the chain. They glob together. Quaternary structure is when you get multiple chains interacting with each other to form an even larger complex. Protein denaturation is when you get unfolding of any of these structures except primary. If you break that chain, it's a new compound entirely and is not considered denaturation. When you put the protein in a new environment, it can shift slightly, which is called conformational adaptability, and is not considered denaturation. But if you get a large unfolding of this protein, where this shifts over completely, or this unfolds from its beta sheet state, it's considered denaturation. This denaturation is reversible sometimes if you remove the denaturant, such as temperature or pH. It can also be irreversible. Most of the times it is reversible if the protein is small and is in small concentrations. Otherwise, you get pieces interacting with each other that should not interact. There are 2 main forms of denaturation in industry: temperature and pH. Temperature is usually done through increasing the temperature, as when you increase the temperature, hydrophilic bonds overexcite. As they excite, they break, and you get unfolding of those bonds that were formed via hydrogen bonds. pH is a little bit different because in the center you have the most stable at the pi of the protein, but at the extremes you get ionization of hydrophobic portions of the protein. Hydrophobic portions are usually in the middle of the protein and don't interact with the environment. But when they get ionized, they try to push towards the outside so they can interact with the environment. This causes a whole change in the protein structure and is a large denaturation. These denaturations are important in food as they affect a lot of properties that both customers and industry is concerned with. Enzyme activity is one such property. Proteins are often enzymes in food compounds, and if you do not inactivate them, they can make the shelf life bad of the product and make it not last very long. So by denaturing these, by either raising the temperature and destroying the protein structure prior to packaging, or raising the pH of the structure, raising the pH of the food throughout the entire time it's going to be in its container, you can prevent this enzymatic activity to where it won't make the product go bad. Emulsifying is also a good thing that can be done via protein denaturation, as when it's in its natural state, most hydrophilic bonds are on the outside, not many hydrophobic. But when you denature it, you get some hydrophobic portions on the outside that can interact with oil-based products, and the hydrophilic portions that are still there can interact with the water portions, making it to where they won't separate into 2 layers. But this also affects solubility, as since the water is going to be in most products, it won't be as soluble anymore because you're going to have those hydrophobic portions sticking out. Digestibility is also affected when you denature proteins, as just slight unfolding can make it easier to reach for our enzymes in the stomach, but too much denaturation and twisting can make it to where our enzymes can't attach. Also, overheating of proteins can cause hardening, which will make it to where our enzymes can't really digest it well and make it nutritionally unavailable. This concludes my discussion on protein denaturation.
Hello, Dr. Lana. It's Andres, and today I will be speaking about some important reactions regarding sugar. Most reactions in sugars involve carbohydrates, Although there are many, I've narrowed them down to 5. And so today I would like to talk about first is browning reactions, which include caramelization and Maillard reactions. Maillard reactions is a chemical reaction between the amino group and the reducing sugar to produce this browning effect. It usually occurs anywhere from 140 degrees Celsius to 165 degrees Celsius, and it's non-enzymatic. The carbonyl group of the sugar reacts with the free amino group of the acid in the amino group. So essentially we have the reducing sugar with the enzymatic browning, and then we have the free amino group and the water to produce this browning effect. At higher temperatures, greater than 165 degrees Celsius, we have caramelization. So basically the reaction rate depends on temperature, on pH. If the pH is less than 5, Maillard reaction can occur, but if you want to inhibit Maillard reaction, one of the ways is to increase the pH of the reducing sugar above 5. And other ways to inhibit the Maillard reaction is to control the moisture, control temperature, and lastly, you can block the substrate to inhibit these reactions. Results in browning can cause a flavor alteration. It can reduce nutrient value in the product. It can also have mutagenic effects with the products, and this can be harmful when ingested, and a precipitate may form. So some desirable effects that Maillard reaction have for certain products are bread and coffee. The crust in the bread, the browning, that's desirable, the coffee, the browning. And in some products that it's undesirable are in dry eggs, condensed milk, clear juices, and dry egg whites. And that results in a formation of a dark pigment called a melanoidin. And that is a disadvantage because consumers do not want that. So out of the main reactions, Maillard was my first one, and my second one is substitution. So an example where substitution is used to produce a sugar would be where alcohol groups are substituted with amines, or where alcohol groups are substituted with hydrogen. We have a formation of an amino sugar where the alcohol group in a glucose is substituted with an amine to produce a glucose amine. Second, we have an example of the formation of deoxy sugar. And a good example of that is where we substitute the hydroxy group in a deformed ribose with a hydrogen to produce a 2-deoxy-D-ribose, which is a deoxy sugar. So third reaction that I want to talk about today is hydrolysis reaction, which involves a cleavage of chemical bonds with the addition of water. A good example of this is in the production of ascorbic acid when starch is hydrolyzed to make glucose. So hydrolysis occurs when a carbohydrate is broken down into sugar molecules. A good example is when sucrose is broken down into glucose and fructose. Both weak and strong acids can undergo hydrolysis, and some hydrolysis examples, besides involving weak and strong acids, are acid-catalyzed nucleophilic reactions, the protonation of alcohol groups, and then the addition of water to release a proton. So fourth, we have reducing sugars, which produce a polyhydroxyl alcohols. Some of these alcohol sugars are mannitol, xylitol, and glucitol. Formations of sugars can be done by hydrogenation, and usually these sugar alcohols are less sweet and have some or the same caloric value as regular sugar. And they actually do not promote sugar decay, so that's a benefit. Lastly, we have oxidation reactions. These include the oxidation of aldehydes and or hydroxy groups in sugars to produce certain acids. For example, we have the oxidation of aldonic acid at carbon 1 to produce gluconic acid. We have the oxidation of uronic acid at carbon 6 to produce glucuronic acid. And lastly, we have the oxidation of alderic acid at both carbons 1 and 6 to produce glutaric acid. And some other main reactions that include oxidation are Fehling's test and Benedict's test.
Hello, I'm going to talk a little bit about leavening agents. And leavening agents are used when wanting to form a nice, airy, fluffy texture or just simply a rise in your product. And there are three leavening agents that I would like to talk a little bit about. First off, there's a mechanical leavening agent, then there's also a biological leavening agent, and then there's also a chemical leavening agent. So mechanical leavening agent occurs when, for example, you're whipping up egg whites, and the proteins in the egg whites actually open up a little bit more so that they can attach and trap in more air bubbles. And so it causes a nice foamy texture, which a lot of people like to use to make their product more airy and fluffy. For biological leavening agent, we're going to talk a little bit about yeast. Yeast is a living organism. It's actually a fungus, if you did not know. And there are two different types of yeast. There's more of a normal type of yeast, which just mainly eats sucrose. And then there's also a faster special yeast that eats sucrose and maltose at the same time. So when these yeast organisms, when they eat sugar, in the presence of sugar and water, they create carbon dioxide as a product, and these carbon dioxide bubbles actually build up and they again cause a rise in your product. This reaction can actually be sped up due to temperatures. They have an optimal temperature, and so when you are waiting for your bread to rise, it's better to keep it at somewhat of a warmer temperature so that the yeast can actually have a faster rate of reaction. And we're going to talk now, switch gears to chemical leavening agents. For example, you have baking soda and you have baking powder. And so baking soda is sodium bicarbonate, and in order to cause a reaction, you have to add some sort of acid. So for example, sodium bicarbonate is going to go in this jar right here, and then I have citric acid right here. And so when you put them together, they cause these bubbles as you see here. And so these bubbles are useful for your product, as I have been saying, to cause a rise or a fluffy texture. These acids vary in reaction rate depending on different properties, such as dissociation properties. Solubility, for example, is a main one, and also whether or not it's coated in an alkali metal. So for example, a fast reacting acid would be monocalcium phosphate as opposed to a slow reacting acid, which would be sodium aluminum phosphate. So again, as I said, monocalcium phosphate has a more soluble nature, and while as a slow reacting acid, the sodium aluminum phosphate is coated with the aluminum alkali. So that causes a slower reaction because it is slower to dissolve into your substance. And so now we're going to talk a little bit about baking powder, just for a little bit. So baking powder is essentially baking soda, but the thing is, you don't have to add acid to your baking powder. The acid's actually already added into it. So it's sodium bicarbonate with an acid in here in a dry form, and it's stable because it's dry. in order to cause this reaction, you actually have to add water so that the ions can separate and then find each other in this water solution and then cause carbon dioxide to form, and that is baking powder.
Hello, Dr. Lana. It's Andres again, and for my second presentation, I will be speaking on grains. Now, grains are small hard seeds that are with or without fruit layers, and they're harvested primarily for human consumption or for animal feed or animal consumption. Now, they're a hot topic of research because in today's world, many scientists are looking for alternate ways in order to produce fuels. Corn is being looked at in order to produce or being used as a biofuel to replace alternate fueling. Now, crops are divided into two types. The first type are cereal grains, which include wheat, corn, rice, barley, and rye. And then our second type of grains, we have legumes. Legumes such as soybeans and beans. Now, after harvest, the dry grains have certain advantages over other staple foods in that they're easy to handle in regards to transportation and storage. These dry grains, they can be stored at room temperature for long periods of time and be fine. There's no need to cool them or heat them when transporting, so that saves money. And this is due to the composition, and I will discuss that in a few minutes. Grains are made up of complex carbs and proteins and are considered a major caloric value worldwide. And grains are around 12 to 14% in water activity. Now, all grains may look the same, but they may look different, but they're actually they are made up of the same characteristics, which include so here you have your grain, and then you have your bran, which constitutes to 17% of the weight, and then you have your germ, which is right here, and that constitutes to about 3% of the weight for the grain. And then right here you have your endosperm, which constitutes to around 80% of the weight of the actual grain. Now, the bran protects the grain. The inner portion of the bran is high in niacin, and the actual bran is a good source of dietary fiber, which includes cellulose. And also it has minerals such as magnesium, phosphate, and calcium. Then we have the endosperm, like I said earlier, which constitutes for 80% of the weight, which is composed of cells containing starch. The limiting amino acid in the endosperm is lysine. It's also a good source of riboflavin and minerals. Lastly, we have the germ, which is made up of carbohydrates, lipids, B vitamins, and enzymes and tocopherols. Actually, the germ can be processed to make flour and cereal breakfasts. If the germ or if the grain germinates, it produces a variety of enzymes which are actually beneficial in the production of beer, but disadvantageous or not wanted when producing cereals and other products, for example, flour as well. Lastly, when the plants process or make these seeds or cereal grains. They're naturally converted from glucose. They're converted from starch to glucose to have a low water activity. And this low water activity is essential because when it germinates, a low water activity will provide a higher yield in enzyme activity and thus germinates faster.
Hey, Dr. Lana. So I'm going to talk to you about proteolytic enzymes. Proteolytic enzymes is any enzyme that hydrolyzes the peptide bonds that exist between amino acid residues and various peptides and proteins. Proteolytic enzymes are used in the food industry for producing desirable changes and alterations of flavor and texture. The most common example would be rennet, which, as you know, is used for cheese making, and then, of course, papain, which is taken from papaya and used as a meat tenderizer. Other examples of proteolytic enzymes used in food production is, for example, would be tofu, soy sauce, dry aged beef, where the beef is allowed to tenderize, and then sometimes even to keep from proteins from precipitating out of beer. Proteolytic enzymes are used in that as well. So now that we know what they are and how they're used, we'll talk a little bit about how they work. So it's important to understand what a scissol bond is. And the scissol bond is described as the unique sequence of amino acids on the substrate that is required for the enzyme to register that sequence and to hydrolyze a particular bond. So the scissol bond is where the reaction occurs, but at places other than the actual cleft site where the reaction occurs, other amino acids are going to affect the affinity of the reaction and then actually if it'll proceed. For example, rennet is highly specific in hydrolyzing the bond between amino acids number 105 and 106, which are phenylalanine and methionine residues. And it's highly specific for hydrolyzing that bond, which is present on the alpha-casein protein that keeps the casein micelles dispersed in solution. So when it hydrolyzes that bond, it kind of gives the casein micelle a haircut, and that allows the casein micelles to aggregate together, and then the cheese process continues from there. Papain is an example of an enzyme that shows more specificity for places other than the active site. So it's actually amino acid residues that are going to affect the affinity of the reaction. For example, proline, which, as you know, proline, which is shaped like this, is going to provide steric hindrance in the papain enzyme. Histidine residues, which are positively charged, are also going to react at the active site and provide the electrostatic forces necessary for the reaction to proceed. So those are just two examples of how the reactions occur and how the amino acids that are present on those peptides are interfering or helping with the reaction. So lastly, now that we understand a little bit about the rennet and the papain, so rennet is derived from the stomach of young calves, and obviously there's a problem with producing enough of that as cheese production continues to increase. There's not enough calves to provide the rennet. So what they're trying to do is produce microbial versions of the enzyme, but the problem is that these microbial versions of the enzyme don't have the same specificity for amino acids at both where the reaction proceeds and at places that are distant from that location. So this leads to the problem of the rennet continuing to potentially break down the amino acids and provide undesirable flavors, or this lessens the effectiveness of the enzyme relative to the natural chymosin, aka rennet, that would be traditionally used in cheese making, especially in Europe where it has to be used. So this concludes my discussion on proteolytic enzymes. Thanks.
Awesome.